Hey everyone, hope you're all doing well. Happy holidays. Today I want to talk about aging. It's a topic that scientists as well as huge companies are never shy to discuss, and it's obvious that in our age of finding the fountain of youth and keys to longevity, we've spawned a pretty big era of intense bioengineering. Companies like Google have pumped millions and millions of dollars into startups that promise life extension, but it's also important to realize that smaller labs have actually taken the lead on this very serious issue, like the ETH researchers in Zurich. They've isolated the genes that actually modulate the aging process, and where identifying the genes is a key component of halting, if not completely reversing, our body's natural mechanism for aging, it's definitely something to discuss. So let's check it out. So how does one actually go about finding the genes responsible for aging? It seems that with the massive complexity of the human genome, things start to get a little hazy, but the solution might actually surprise you with its almost brute force way of looking for the proper genes. The scientists chose 3 animals, all with vastly different biochemistry, and kept looking for a common set of genes that eventually resulted in aging across all 3. While the choice of animals were a nematode, a zebrafish, and a mouse, it seems pretty basic on the evolutionary scale, but trust me, it was an entirely different challenge when they were actually looking for the gene. The scientists liken the process to finding a needle in a haystack of over 40,000 genes. It's pretty interesting and challenging at the same time. So I think that before we can talk about the study and its ultimate findings, it's important to discuss the basics of genes, what they are, and how the expression of such traits is actually measured. Genes are stored in these long, windy things called chromosomes, and they encode all of your biological information, whether it be your eye color, muscle mass, or really any features of ours that we often take for granted. A measure of gene expression can be found in the number of mRNA molecules present within an animal's cells. When the mRNA of a specific gene is widely apparent, it means that the physical or internal attributes of that gene are very evident. However, if there's not much of a trace of the gene's mRNA, then understandably it means that the gene expression is minimal. The researchers used algorithmic reasoning and statistical models to try and map the intersections of genes that were expressed in similar manners across all 3 species. Now, if you think about it, across widely different animals, there are only a few forms of gene expression that could be similar, such as aging. Of the 40,000 genes in their search, a mere 30 became of interest. One in particular, the BCAT1 gene, seemed to have an overwhelming effect on aging. When its effect was clouded by certain enzymes and other measures, it extended the mean lifespan of the nematode by up to 25%. The gene actually regulates an enzyme that degrades certain amino acids that are naturally occurring. The interesting bit comes when the gene's control is turned off, meaning that these amino acids are free to populate within a cell, and that creates nifty benefits like longevity and, in some cases, extra speed. The greatest thing is that all of these benefits can be available to humans because the scientists looked exclusively at genes that were conserved in evolution and thus available to all organisms. While this doesn't mean that you'll live forever, the certainty of life extension is definitely something to consider. We've come a long way, and our generation is often told that we're in for some serious advancement in the field of medicine, and we don't quite believe it until technology like this rolls around and rebolsters our faith. While a small test of amino acids and gene expression might not tell everything like it is, and there's no telling to what the dangers of such a technology might be, it's nonetheless a revolutionary form of science that I, as well as a lot of people, are excited to see. And as always, thanks for watching, and I'll see you guys next time.